In the previous two video lectures, we understood the cell cycle and cell cycle checkpoints. Today, we will study the details of the cell cycle control system. Let's begin. We know that cell division is crucial for normal growth, development, and maintenance. But, in the body of a eukaryotic organism such as human, the rate and timing of cell division varies with the type of cell. For example, skin cells divide frequently throughout life and specialized cells such as nerve cells do not divide at all. The reason behind these differences in the frequency of cell division is the regulation of the cell cycle at the molecular level. The cell cycle is regulated by external and internal signals. In other words, the cell cycle control system is made up of external signals received by cell and its internal environment. Let's first find out what are these external signals. We understand that cells of the human body communicate with each other. This cell-cell communication is part of regulation and coordination system. So, cells communicate, signal each other and thus convey what to do. These signals or signaling molecules that regulate and coordinate cell proliferation throughout the body are known as growth factors. Now, growth factors can either inhibit or stimulate cell proliferation. For example, protein called epidermal growth factor is secreted by endocrine cells. Epidermal growth factor stimulates epidermal cells such as skin cells to divide. So, at this point of our lecture, it is clear to us that growth factors signal a cell whether to proliferate or not. Now the next question is, how these growth factors regulate cell proliferation. Let's say, this is a cell that secretes a growth factor. Once released, this growth factor acts on a nearby cell. The nearby cell has a growth factor receptor on its surface. So, growth factor binds to this receptor. Here, note that growth factors act in short range. And this kind of signaling is known as paracrine signaling. This binding stimulates a signal transduction cascade inside this cell. Now, what is signal transduction cascade? It is a series of biochemical reactions that convey the signal from outside of the cell to its nucleus. So, now the signal has reached the nucleus. Inside the nucleus, this signal activates genes that encode transcription factors. Transcription factors are proteins that control the rate of transcription of genetic information. Now question is, how? By regulating another set of genes. Transcription factors either activate or repress the expression of these specific genes. Here comes the another question, what kind of genes are these? These genes are those whose products will either promote cell division or inhibit cell division. In other words, we can say that transcription factors regulate cell cycle genes. Most important of these cell cycle genes are the genes transcribing cyclins and cyclin-dependent kinases. We will study them shortly. I hope now you have a clear idea about the first part of the cell cycle control system. That is, the extracellular signals that regulate the cell cycle. Let's now understand the second part, the internal environment of the cell. There are two types of cell cycle regulatory molecules present inside the cell. These are protein kinases and cyclins. Let's understand them one by one. Protein kinases. Protein kinases are enzymes that activate or inactivate other proteins. They do this by phosphorylation. Kinases are present at a constant concentration in a growing cell. but much of the time they are present in the inactive form. Kinases become active only when they associate with cyclins. Cyclin is again a protein, and it is named so because its concentration fluctuates cyclically in the cell. As we said before, for the activity of kinases, their association with cyclins is a must. And because of this, these kinases are known as cyclin-dependent kinases or cdks. As the concentration of cyclins varies, the activity of cdk also varies. Let's understand this concept by illustration. Let's say this is a cyclin-dependent kinase. It combines with a cyclin, and thus a cyclin-CDK complex is formed. As a result, this CDK has now acquired the ability to phosphorylate target protein. As we said before, cdks can either activate or inactivate the target protein. Suppose this is the target protein present in active form. The cyclin-CDK complex phosphorylates this protein and the protein becomes inactive. Similarly, if the target protein is inactive, cyclin-CDK complex phosphorylates it and protein becomes active. So, now we understand that cyclins and cyclin-dependent kinase are the proteins that are responsible for guiding a cell from one phase of the cell cycle to the next. Let's now summarize today's lecture by understanding how external and internal signals together work in a cell cycle control system. Suppose this is a cell, and this is a growth factor receptor. First, a growth factor binds to this receptor. Through the signal transduction cascade, the cyclins are synthesized. This is a cyclin-dependent kinase. In the next step, the cyclin binds to this CDK and CDK get activated. Activated CDK further stimulates activities that are required for cell division. For example, synthesis of proteins required for DNA synthesis, stimulation of DNA replication, chromosome condensation, etc. I hope you have now understood basic concept and components of cell cycle control system. Thank you for watching.